In a previous video, we discussed the structure and basic function of DNA. In this video, we're going to do a comparison between DNA and another nucleic acid called RNA. So here's a picture of both RNA and DNA. The sugar found in DNA is deoxyribose. DNA is double-stranded. DNA contains thymine, which is the T. You can see that right here. DNA is staying in the nucleus of eukaryotic organisms. So if it has a nucleus, DNA is always going to be inside of it. And DNA is the original copy of the information. Now, how does this contrast to a RNA molecule? Well, the sugar in RNA is ribose. RNA is typically single-stranded. RNA contains a base called uracil, so we can see that up here. And if we look at the bases, CGA in RNA are the same as CGA in DNA, but there is a change when it comes to the last base. In DNA, we have thymine, the T, but in RNA, you only find what's called uracil, which is the U. RNA can leave the nucleus, so it's able to be made in the nucleus and then leave and go out into the cytoplasm. RNA is considered a copy because it is created by DNA in order to leave the nucleus with the information to go make proteins. Now we have what's called the central dogma of biology. The central dogma of biology basically says that DNA will be converted into RNA, which will then be converted into a protein. And we know that genes are found in DNA and they create our traits. Well, one of the things that we are going to learn is that proteins actually create the traits that we have. And so DNA, the code, gets converted into RNA, which eventually gets created into proteins. So the code is passed from the DNA to the proteins. And that's how we get our traits. Now, a couple things about this picture. The process of DNA going to RNA is called transcription. And the process of RNA being converted into a protein is called translation. And if we wanted to make more DNA, that would be the replication process. But this video is going to focus on DNA being transcribed into RNA. When we talk about the central dogma of biology, we typically think of it as what's called the unquestionable truth. This is the primary way things happen. DNA is converted to RNA and RNA is converted to protein. So it states that the information that is found in DNA is converted into RNA and finally to a protein. We can see that DNA replication would happen in the nucleus, and we can also see that DNA transcription to RNA was also going to happen in the nucleus. And then finally, translation would happen in the cytoplasm. But we're going to focus again on DNA being converted into RNA in the nucleus. There are three types of RNA that we are going to eventually talk about. One is called messenger RNA, also known as mRNA, and that brings information from DNA that's in the nucleus to the ribosome. So it's carrying the message. We can see that right here would be a little example of an mRNA molecule. There's something called tRNA, which brings amino acid, those are our protein building monomers, to a ribosome so that the protein can be made. And we can see over here that this would be a tRNA molecule carrying an amino acid. So you put a bunch of amino acids together, you get a protein. And finally, we have another RNA molecule called rRNA. That's a ribosomal RNA. These are the building blocks of ribosomes. So there's a lot of rRNA found in a ribosome that will then be making the proteins from the information found on mRNA by using amino acids that are brought in by tRNA. So again, DNA is going to get converted into RNA, which gets converted into a protein. We're focusing on transcription, which is DNA to RNA. So how does it work? Well, transcription is basically opening up DNA and then complementary base pairing free bases to be put together into an mRNA strand. We do this with an enzyme called RNA polymerase. RNA polymerase. ACE for enzyme. ACE is generally an ending suffix for enzymes. We're using an enzyme to create a polymer of RNA. So RNA polymerase opens up the DNA helix. It starts putting RNA bases together into an mRNA strand. And remember, there's no thymine, there's no T's in RNA. There's U's instead. So here I see a T in the DNA. Therefore, the RNA is going to be an A. Here I see an A in DNA. That means its complement is going to be a U because there's no T's in RNA. G always matches up with C, so that doesn't change. But anywhere I have an A on my DNA, then its complement from RNA is going to be a U. And then eventually it all gets stuck together, and then the mRNA will leave the nucleus to go make that protein. But right here in the transcription process, we are taking information from DNA, from a gene, and we're making it into an mRNA strand that is allowed to leave the nucleus. On mRNA, we find these 3-letter codes. Basically, every 3-letter codes is called a codon. Codon. Here's a codon, AUG. That's one codon. AAC. That's another codon. A codon is a group of 3 nucleotides on messenger RNA that specify a particular amino acid. So these 3-letter codes tell us what amino acid is going to be put in that space by the ribosome. The first step in transcription is that DNA unzips. It unwinds to open up the gene so that mRNA can be made. The second part is new RNA bases will attach to one side only. So we can see that once the DNA is opened up by RNA polymerase, the new bases come in and match complementary to the DNA. And remember, there is no T's in RNA. You will only find the U's. Then we have the newly copied strand of RNA is going to leave the nucleus to create a protein.